Cancers, through their evolution, have acquired a number of ways of evading the normal mechanisms that keep them from forming. And through that, they acquire mechanisms to adapt very easily. And that also allows them to adapt to our therapies and become resistant to them, and to go into new places and metastasize from the tissue or organ from which they originated to a new place. So it makes it very challenging for treatment when cancers develop and acquire these properties that allow them to adapt. Our lab is very interested in understanding how mutations in particular genes cause cancer. What we've also learned is that mutations alone are often insufficient to promote the development of cancer, arguing that there are additional factors that may play a role. Some of these factors may actually be in our environment or within ourselves. And this includes the foods that we eat, the microbes that are within our body, and even the other things that we're exposed to in the environment. And so our lab is very interested in studying how these mutations in cancer genes cooperate with environmental factors and host factors such as diet and obesity to promote the development of some of the most lethal cancers, in particular lung and pancreatic cancer. We use a number of genetic engineering approaches, including CRISPR and DNA recombination, in various models of cancer that allow us to recapitulate the events that occur in human cancers in these models and understand how tumors evolve. We also use a number of methods to analyze these tumors as they develop, including the genes that they express using RNA sequencing, including at the single cell level. We use DNA sequencing to observe additional mutations that are acquired. We use metabolomics, a method to look at how cells rewire their metabolism as cancers develop. And we use proteomics, which are methods to look at how proteins function and how cells signal to cause cancer. And finally, we use a number of imaging-based approaches, including fluorescence-based imaging, that allows us to observe how cancers evolve from their various earliest stages all the way to full-blown advanced disease. Our hope is that by understanding how cancers form and develop, we can identify new approaches, new strategies to prevent, intercept, or treat cancer. By understanding the basic mechanisms and the biology of cancer development, we can determine how we can modulate not just with drugs and new therapies, but also perhaps changes in our diet or other factors that we can use to prevent or treat cancer better.